that. And she.
What you do?
Okay.
All right. All right, guys, if you're just coming in, you can come up and grab your exam and your Scantron. If you leave before class is over, though, you can keep your Scantron. Please give me back the exam. We'll go over it. Yeah, if you leave before the other class, please give it back. Also, you guys have your data analysis due today. So remember, that's one copy per group. Be sure everybody's name is on it so they get credit for it. Turn that in up here as well.
Grandma was on the phone. I sent it to my 13-year-old, but I might be on help. Too young to have it. My drink is not. I think it's.
Vote for me, thank you.
She's really focused there.
Yeah, if you go into the one that you're, we can help, it says, the QR code.
It's such a good one. Yeah, what are the decisions video vision, it's fine. I think so too.
I think that's what mine is.
What did it say? It's fine, computer. Oh, well, you really want to get pharmacy, you really want to get pharmacy.
So if you're just coming in, please come up and grab your exam and your Scantron. And if you have an in-class data analysis to turn in, please turn it in up here as well.
Just gonna ask, cause mine's in the car over then.
You can, or you can get it when we take a break. It's up to you.
Okay, sounds good. I'll do it. So anyway, what were you saying? What is nothing? I know somebody that just got accepted at USF with a 2.8. So what? It's fine. It's not just, it's fine.
That's not much better than this. It's gonna, don't think about it.
I'll do it.
Just the other good things.
If you guys only have three in your group, that's
For me versus
Yeah.
the number, you know.
okay.
Person.
Again, guys, be sure everyone's name is on that list, including the author, whoever wrote it up. Miss your name,
As the author.
whoever was the
Yeah.
leader or whatever team leader.
Yeah.
So if you didn't grab your exam, be sure you grab your exam and your Scantron. If you leave early today, please give me back the exam, okay? You can keep the Scantron, but I'm gonna collect the exams. How's everyone doing?
Oh, yeah, by the way, I'm a grandma.
Which is better than being a parent. I'll tell you. So, all right, just a little announcement about Monday. So we're doing the Rubisco lab. So the write-up for that lab is it's pretty lengthy. I'm not going to be able to make photocopies for you guys and bring those to class. However, I do have like a quick guide that I'll be able to put up on the projector, and that will be helpful. But the lab is kind of involved, and we're going to spread it out over 2 weeks, and then actually on the third week you're going to do the analysis. So it may be helpful to you to have a copy of that with you. You know, that's completely up to you. It's just a kind suggestion, right? That lab should work. I don't know what happened with the peroxidase lab. That was pretty regretful, but my guess is that something with the peroxide solutions might have been not good. So peroxide is pretty volatile, so it will oxidize also quickly just if it's not stored correctly. So I wonder if that's just an old bottle of peroxide that we had. But we're going to troubleshoot it, hopefully get it fixed for next semester. So did everyone who didn't get a brown color when you guys mixed the first two? Oh, is that one group or is that?
Yeah.
Okay. All right. Because that tells me, what does that tell you if you get a brown color in the first part? That the enzyme's working, the extract's working. There's peroxide there to be broken down, and also the detection reagent is working. But why we were getting no signal on the spec, I haven't fully figured that out yet. So it's not a very complex exercise. But the Rubisco lab that we're doing on Monday, I've done that many times, so we should get some good results from that. What is Rubisco? Who knows? One of the most abundant proteins on Earth, right? So it's one of the first proteins involved in the Calvin cycle, which is like a big part of photosynthesis, right? So we're not going to talk much about photosynthesis in the lecture, but this is our opportunity to kind of touch on a little bit in the lab. We're going to definitely get into aerobic metabolism, mitochondrial respiration, but some of that photosynthesis stuff I kind of skip over. All right, any general questions or anything? Moving in a good direction here. All right, so hopefully everyone checked out the previous lecture, which remember we had just available online. Today's lecture will be recorded as well and posted. So what I'd like to do today is just get through the first of 3 sections that I want to cover today, which is DNA repair. We'll take a little break and we'll go over exam 1, and then we'll wrap up with talking about transcription, both prokaryotic and eukaryotic transcription, and finally wrap up looking at RNA processing. So the process of transcription, you guys definitely had that in Bio 1, but we're going to definitely take those processes up a notch, a couple notches, learn about a lot more proteins and transcription factors that are involved. So I think you're going to find that a lot of this material is starting to become new. And especially in this chapter, it's unlikely that you've had most of these, because even in genetics, if you've taken that course, we don't really cover a lot of these mechanisms like we cover here today, right? So just keep that in mind moving forward. We're going to be having a lot more new material. All right, so let's dive in here. So section 5.2 has to do with DNA repair. So damage to a cell's DNA can be anything from minor to completely catastrophic, right? So DNA polymerase has relatively okay fidelity. What does that mean? Fidelity. Yeah, the accuracy of it. It's okay. You know, when you do a PCR reaction, which is basically in vitro replication without proofreading enzymes, you know, you run into a mistake every about 1,000 base pairs. So that would be extremely significant to the integrity of the cell genome. We introduce way too many errors. You know, all of our children would be extreme mutants. If that was the case. So life has evolved a very elegant system of repairing those mistakes. It's almost expected that those mistakes are going to occur. And a lot of the mechanisms have really become clear in the last 15, 20 years. Okay. All right. So keep in mind the DNA in the genome is a permanent copy, right? So, severe consequences to damage. What's one that comes to mind? Possible side of, what is that? Well, that would be a cause. What would be a result of cell mutation, genomic mutation? Cancer. Right? It's a big one. So, types of DNA damage, and that's what we're talking about here, DNA damage, not mistakes with polymerase, include like a spontaneous form of damage. This is a specific one that's characterized where you deaminate cytosine. So you take off the nitrogen group and it basically becomes like a uracil. So it's a uracil analog. UV radiation as well. So ultraviolet light causes a very specific type of mutation called a pyrimidine dimer. So this is most common when you have two T's adjacent in the genetic code, and they're energized with ultraviolet light, and they end up joining together through this cyclobutane ring that forms. So now when DNA polymerase approaches that dimer, it doesn't know what to do with it. It's this big bulky molecule. And then there's chemical carcinogens as well. So chemical carcinogens specifically add these big bulky groups on top or attach it to the DNA covalently. And that can block transcription, it can block replication, cause a lot of problems. So, these things, like these causes for damaging DNA, have been on the planet for a long time. And life has evolved some ways to overcome it. So the top there, you can see cytosine being deaminated, it becomes a uracil. You can see what the cyclobutane ring looks like when two T's are together and they get polarized with UV light. And carcinogens specifically will add these, like what are called adducts, or big bulky groups to a specific nitrogenous base. So it can happen to any of the four bases. They're just showing you guanine here, giving this bulky group addition. All right, so there's basically two classes of DNA repair systems. So you have a direct reversal system, which is very specific. So it reverses whatever chemical reaction caused the problem in the first place. And then you have what's characterized as excision repair. So this is actually removing the problem from the DNA and then replacing it with the correct nucleotides. All right, so remember that the nucleotide is made up of a sugar, phosphate, and what's the other part? Nitrogenous base, right? So DNA can either damage the base, it can damage the sugar. It just depends. All right, so one example of direct reversal is something that happens in many eukaryotes, but interestingly, not in humans, not in mammals. So what has evolved is whenever cells detect these pyrimidine dimers, so these two T's side by side, they have another mechanism that involves this enzyme photolyase that requires UV light to activate it. So here you've got DNA damage that was caused by UV light, and you've got the solution that also requires UV light activation. So, this is real prevalent in yeast, in bacteria, but not in mammals, interestingly enough. So mammals have come up with an alternate way to overcome pyrimidine dimer mutations. and that is excision repair. So instead of going and directly fixing it with the specific enzyme, we're just going to cut it out and then replace it with the correct nucleotides. All right, so there's a couple types of excision repair we're going to talk about. One is base excision repair. All right, so one specific mechanism of this involves this DNA glycosylase. So DNA glycosylase will come along, it will remove the specific incorrect base, and it creates a specific type of site called an AP site, which stands for apurinic or apyrimidinic site. The next step is a endonuclease comes along and cuts up the backbone of the AP site. And then these next two steps you're going to see are very consistent with all of these excision repair mechanisms. You get a new DNA polymerase, right? Not DNA polymerase epsilon, which is coming in and building the DNA, but a new type of DNA polymerase that comes in and replaces the nucleotides, and then DNA ligase seals the nick. All right, so this mechanism using DNA glycosylase is very specific for that cysteine deamination, which we were talking about, where this cytosine becomes a uracil. All right, so you can see up here, we've got a U instead of a C because it's been deaminated. So the enzyme DNA glycosylase comes in and it just removes the nitrogen and base. So we call that an AP site that does that. Right, so the reason that's important is because it takes that apurinic or apyrimidic site to be recognized by an AP endonuclease. So what is the difference between an exonuclease and an endonuclease? So endonucleases cut within the sequence, and exonucleases can only start eating away nucleotides outside the sequence, so at the ends, specifically the 5' and the 3' ends. All right, so AP endonuclease comes in and nicks the DNA, and then there's another enzyme that will fully pull it out, because remember, this is base excision repair. And then DNA polymerase and DNA ligase come in and replace the nucleotide with the correct one, and DNA ligase seals that nick, right? DNA ligase can create a phosphodiester bond. All right, so that's base excision repair. Nucleotide excision repair is more prominent in carcinogenic or mutations caused by carcinogens. So because it has to pull out that whole nucleotide that has that bulky addition group. So one important comparison, though, when you're looking at nucleotide excision compared to base excision, is with nucleotide excision, you have to remove a lot of nucleotides. So you'll remove the correct nucleotides that are, 5' and 3' of the specific mistake. There's a good system that's been recognized or identified in bacteria that uses these UVR ABC endonuclease proteins. All right, so what's nice about this mechanism, it's pretty easy to remember because it just goes in order ABC. The first step is UVRA. It recognizes the bulge or the inversion in the DNA double helix, right? So if you have a mispair, a mismatch, remember, you're going to have, so in normal DNA, how many rings across should the molecule be from one strand to the other? So remember, purines have 2 rings, pyrimidines have 1 ring. So the diameter of a DNA molecule should be 3 rings. If you have a mismatch, it's either going to be 4 rings or 2 rings. So the shape of the DNA molecule is going to be not like this, but either like this or like this. Okay, so this would be 4 rings across. This would be 2 rings across. There are proteins that can detect that. Right? So these proofreading proteins, such as UVRA, can detect that. Right? So it finds the mistake, and then the next 2 proteins, UVRB and UVRC, cut out like a 15 nucleotide size fragment, that includes the mistake. And then a lot of these steps, you're going to see this common among all of the repair mechanisms, and they'll start to make sense, I think, as you read over them more and more, because they're necessary to repair DNA. You have to unwind it a little bit, right? You have to remove the nucleotide. Only helicases can do that. You have to fill in the gap. You use a polymerase to do that. And then you have to seal the nick and create that phosphodiester bond to complete the molecule. All right, so here's an example of nucleotide excision repair. This is a pyrimidine dimer. We have 2 T's. And whereas like a replacement repair would just remove the mistake, you end up excising an entire oligonucleotide, right? So oligonucleotide, that's an important term if you don't know it already. It's just many bases together. It's a short fragment of DNA, anywhere between like 15 and 25 base pairs. That's what an oligonucleotide is. After you remove the oligonucleotide, you then replace it using DNA polymerase, and you seal the nick with DNA ligase. All right, so that is the system in E. coli. It uses those UVR proteins. The system in humans was discovered a little differently. So this condition, xeroderma pigmentosum, is people that have this genetic disorder are very, very sensitive to UV light. Will generate lesions, skin cancers, very, very sensitive to it. And it was a combination of primarily that disease, but a couple others that enabled discovery of how nucleotides were removed in human eukaryotic cells. So it got its name from that xeroderma disease. They called the proteins that did it XP proteins. And just like in the E. coli system, you have to have a protein that recognizes either the bulge or the bending in of the DNA strands. So the first one there is XPC. It'll detect the lesion. And then you get this big assembly, this repair complex. It's like little robots coming in to unwind the DNA, to cut out the oligonucleotide, and then to eventually repair it. All right, so all these processes, and there's a lot of them that have been identified. This is the series of events that has to occur when repairing DNA. You have to recognize it, confirm it, pull it out, build something new, synthesize it, and then ligate the end nick. All right, so you got a lot of proteins here. I don't expect you to memorize these. The only one that I'm kind of stressing here is the XPC, which is the first protein that's involved in recognizing the damage. But then we're going to go over some processes today where you have to start memorizing the names of the proteins and the role that they have in the process. But this is not one of them, so you can note that. All right, but you can see the flow of events: recognition, unwinding, excision, and then repair. Okay, so mismatch repair is a more specific type of mutation that's caused solely by DNA polymerase. So we're not, these are not problems, directly like from DNA damage, either from deamination or UV radiation or ionizing radiation. This is just caused by, the relative fidelity that the DNA polymerases have. It's a natural part of DNA replication. So, but, having either additions or substitutions or deletions occur in the DNA code is potentially very dangerous. So, especially adding or deleting just 1 or 2 nucleotides, because it can cause what we talked about in first couple weeks, which is a frameshift mutation, right? So then you end up getting the completely wrong protein sequence, and the protein is usually inactive in that case. All right, so now one thing that is also unique about these mismatch repairs is in order to do the repair, the cell needs to know which side of the molecule is newly synthesized, right? Because with the mismatch repair, I mean, everything is lining up potentially correctly, but the cell needs to determine what strand has been newly synthesized and what strand doesn't. So a lot of these repair mechanisms are highly active after S phase in the cell cycle. So G2, they see a spike in the numbers of these proteins. I know this is a E. coli example, so it doesn't really have a specific cell cycle, but in the eukaryotic systems, that's very true. So how would a cell be able to determine which strand is newly synthesized and which strand is old? So methylation is a modification that occurs, in the cell. It's a natural component of cell physiology where the bases get methyl groups added to it. And in general, the reason that cells do that is methyl groups will generally silence transcription of that area of the genome. So like within the eukaryotic genome, there are what are called methylated islands, which are large stretches of genomic DNA that have these methyl groups attached. And RNA polymerase is unable to scan that region and builds an mRNA. But that process takes time. Newly synthesized DNA is not going to come out and be immediately methylated. So there are proteins that can detect that methylation status and basically decide and guide other proteins, right, to the non-methylated strand to fix that strand of DNA. All right, so the proteins that do this in E. coli are the MUT, stands for mutant proteins, MUT-S, MUT-L, and MUT-H, three important ones. And they carry, or MUT-S specifically, carries an ability to do strand discrimination. After that happens, fragment is taken out, it's repaired, and then it's ligated. All right, so here you can see in this figure, the top strand is methylated. So that's the old strand. The newly synthesized strand after DNA replication is the unmethylated strand. So the first thing that happens is MUT-S and MUT-L bind to the mismatch base, right? You can see they're targeting it right there because they can detect the bulge in the DNA from having a G and an A, because that's 4 rings apart. And following that, MUT-H will come in. MUT-H can decide or detect which strand is methylated or not. And S and L recruit an endonuclease to come in and cut it out. And then the gap is filled by DNA polymerase. Okay. So, the unique protein here is that MUT-H, which is able to detect the methylated strand and the unmethylated strand. So it removes the correct base. So, what if we remove the base on the original parent strand? How would that mess up would that be sufficient, or would that be, no, because that's the correct base, right? Okay, so in eukaryotes, there is a similar system. These mutant proteins, MUT-S and L, are well conserved in nature. And but they call these MSH and MLH proteins, but they have a similar mechanism. So this process actually is not fully understood. It's an important process, definitely like Nobel Prize worthy. And it doesn't use methylation actually to detect which strand is new and which strand is old. It uses single strand nicks. So one way that you can know this is the lagging strand, right, which is a collection of Okazaki fragments, is going to have more single strand nicks than the leading strand, right, because that is made continuously. So these proteins are able to detect that somehow. They don't know exactly how that works because this whole process, mismatch repair in eukaryotes, is a lot more effective on the lagging strand. So it's hypothesized that it's detecting single or nicks in the DNA. All right, so here you've got DNA replication going on. You've got the leading strand and the lagging strand, and because the lagging strand is produced in short fragments called Okazaki fragments, there are more nicks there that just haven't been filled in by DNA ligase. So these two proteins can locate the nicks, and they can cause the DNA mistake to get repaired. So problem with this is if you end up getting a buildup of errors at this location, the replication fork, you can get a complete collapse of DNA replication at that area. So that's why this is really important in DNA replication. All right, so one of the last things we'll talk about in this section, something called translesion DNA synthesis. So this is specific also at the replication fork. And it uses like different polymerases to work. And it's very important in preventing fork collapse. So let me just go to the figure here. So this uses these specific DNA polymerase proteins like polymerase 5. And you can see here you've got, so this is fixing a pyrimidine dimer as well. So these specific polymerases get targeted right there at the mistake, and these specific polymerases repair the mistake. So the thing with these polymerases and why it's categorized in its own separate category as translesion DNA synthesis is it's not using the original polymerases like epsilon that's generating the primary strands during DNA replication. What happens is when there's a mistake, there's a stall at the replication fork, and then these other polymerases come in to try to fix the repair right away so that the original DNA polymerase can continue to make the new piece of DNA. The problem with these other polymerases is that they have much lower fidelity than the normal polymerases. And so this is it's like a quick rescue during DNA replication if there's an error to get it back up and going. But these DNA polymerases, even though they can fix the error, they carry a lower rate of fidelity. Okay, so probably the most dangerous type of DNA damage is a double strand break. So double strand breaks are typically caused by ionizing radiation, right? So plutonium radiation, things like that. And the reason for that is because they can completely fragment chromosomes, right? It can make the cell almost think that it has polyploidy, so more chromosomes than it should. X-rays and gamma rays are both common causes of double-stranded breaks. If a replication fork collapses because of another type of mismatch problem or something, it can turn into a double-stranded break. And also reactive oxygen species can cause double-stranded breaks as well. All right, so there are two mechanisms that can repair double-stranded breaks. One of them is called non-homologous end joining, and the other is homology-directed repair. So just like translesion DNA synthesis, which uses those other polymerases which aren't as accurate, non-homologous end joining is pretty fast, but it's also very error prone. And it's basically just sealing the broken DNA back together. All right, the other mechanism, which is homology-directed repair, is a lot slower, but it doesn't carry as many errors. All right, so let's look at this first one, non-homologous end joining. So there are these proteins called Ku proteins, and these come after the Ku autoantigen, which was discovered a long time ago, like 60s, 70s. And they will basically bind to DNA at the double strand break. And if the ends are still compatible, it will bring them back together. Right? If that's not possible, there is an additional protein called Artemis, which can actually trim the nucleotide a little bit to make the ends compatible. But yeah, these are pretty unique proteins. There's a lot of them. There's a class of them from Ku70 all the way up to Ku80 that do this. But you can see here you've got ionizing radiation, for example, causing a double-stranded break. The Ku proteins bind to the ends of the breaks, and possibly on their own, they can just ligate it, stick it back together. If modification is needed at the ends to kind of make the ends line up better, there are proteins that do that, such as Artemis. Okay. And then it seals it up, so you get rid of that double-stranded break. So this is a fast and very inaccurate way of repairing the double-stranded break, though, because these proteins, like Artemis, have to usually chew up the DNA a little bit in order to get it back together. So, you know, if that occurs in an exon, then that would be pretty bad. The better way of doing it, but the slower way of repairing double-stranded breaks, uses these proteins that will actually bind to the end of the break, called RAD proteins, RAD. So RAD51 is a protein you'll hear about a lot. We'll talk about it a little later on when we do cell signaling. And it basically coats the end of it. And it actually mediates an invasion into the gene on the homolog of the chromosome. So it'll look for the sequence somewhere else in the genome, essentially get the correct sequence, build a new piece of DNA, and then bring it to the site of the double-stranded break. All right, so let's look at this process. So we've got a double-strand break up here, and this one has really big 3' overhangs. So RAD51 comes and it binds to the overhangs, and it brings it to an undamaged homolog of the gene where the break is occurring, and it ends up forming what's called a D loop. So that's like a homologous recombining of the broken chromosome with the intact chromosome. And so from there, you can either get DNA synthesis of just the broken chromosome alone, or you can get it on both strands and then it recombines. And that's really the take-home message here. So the magic protein of this, and it is a class of proteins, is the RAD51 protein. All right, but this is called homology-directed repair because it requires, you know, the homolog for the gene where the double-stranded break is. All right, because this section has like a lot of different parts, I put together just a little summary slide for you, just the things that we talked about. And for each of these things, usually there was an example that was provided in prokaryotes, an example that was provided in eukaryotes. Okay, so we talked about the damage that can be caused by, or why DNA damage is potentially dangerous. We talked about the two main repair strategies, which is the direct reversal, right? So that's just fixing the problem, or excision repair, which is removing the problem and replacing it. We talked about two types of excision repair, BER and NER. We talked about mismatch repair. We talked about the example in E. coli that uses the MUT class of proteins. And then the eukaryotic example uses the XP proteins. And then we talked about translesion DNA synthesis and also double-stranded breaks, non-homology end joining, and homology-directed repair. And that's it for this section. All right, so why don't you guys take a 2-minute break? We'll go over the exam, and then we'll get into the last 2 sections, which are transcription. How are we doing with this? Pretty nerdy stuff, huh?
Big too.
2025, 2026.
Oh,
Well, the end of 2025.
yeah. I can't even feel because. It's terrible.
Okay. So now we're starting to get into the central dogma of biology, right? The central dogma is going from a DNA code, transcribing that message into an RNA code, and that occurs in the nucleus in eukaryotes, and then going from an RNA code into an amino acid sequence, which occurs at the ribosome in both prokaryotes and eukaryotes. And the first level in the central dogma, they're all important, obviously, but the first level is most important in defining gene expression, right? So remember, all cells in the body have the same genomic blueprint, but why a liver cell is a liver cell and a thymus cell is a thymus cell is because of changes in gene expression. Some genes are on in one cell type and some are off. Some are on or off. Others are on or off in the other cell type. And gene expression occurs at all levels in the central dogma, but at the level of transcription, it's most important and it's most regulated. The more gene regulation you have, the more complexity you can have and variety in different cell types. So just knowing that, you should be able to confer that, well, this process of gene expression or transcription is probably relatively simple in prokaryotes as compared to eukaryotes that have a much more diversity and complexity. And that's very true. The number of factors that are involved in eukaryotic transcription is a lot greater. So first, we're going to cover transcription in bacteria, and but the process as a whole is similar, right? So both require three steps, which are initiation, elongation, and termination. And you're going to see those three steps again in translation when we talk about that. But one thing that the main reason why transcription in eukaryotes is much more complex is the presence of proteins that provides that change in expression, which are called transcription factors. All right, so let's look at bacteria which lack transcription factors, although technically it doesn't, but the molecules are not even called transcription factors. So first, let's talk about the enzyme that actually catalyzes the production of new RNA, producing a messenger RNA, and that is the enzyme RNA polymerase. Right? So, unlike DNA, which requires a primer to catalyze new DNA during DNA replication, RNA polymerase can produce RNA de novo, produce it on its own. And when it's reading the DNA, the double-stranded DNA, it's only reading one side of the DNA molecule. So the side that it's reading is the template strand, and the other side of DNA is called the coding strand because the code is going to be identical to the mRNA that's produced, except you're going to have U's substituted for T's. And that's why it's called the coding strand. All right. RNA polymerase obviously is not using dinucleotide triphosphates, right? It's using ribose triphosphate, so AU, C, and G for that, not AT, C, and G. All right, although it doesn't require a primer, initiation of transcription is going to occur at specific regions along the DNA. It's going to start at specific regions called promoter regions. And it's at the promoter region where regulation is really occurring. Because not much regulation is occurring in prokaryotes, most genes are always on. It's a lot simpler in prokaryotic transcription. All right, the RNA polymerase enzyme is a really large, complex enzyme. It has a lot of subunits. There's 2 alphas, 2 betas, and an omega subunit. And the thing with RNA polymerase in E. coli is it can't recognize the promoter region on its own. It requires an additional factor, which is called sigma factor. So sigma factor works with RNA polymerase by recruiting RNA polymerase to the promoter region after it recognizes the promoter. So there's a larger protein complex called the holoenzyme, which is sigma factor non-covalently associating with RNA polymerase. Right, so RNA polymerase on its own is the core enzyme, but when RNA polymerase is interacting with sigma factor, we call it holoenzyme. Now, I was been kind of insinuating that transcription is not very regulated in E. coli, and that's true, but there are some instances of regulation. We're going to actually talk about that next week more. When we talk about different operons and collections of genes like the lactose operons. The reason for this is it's a waste of energy for the cell to have a gene on if you don't need the product, right? So if there's something not present that gene metabolizes, why spend energy to make it? So because of that, you can do differential expression, but you require different sigma factors to do it. And there's a small family of sigma factors that are present in prokaryotes. For example, sigma 70 keeps the housekeeping genes on, right? Housekeeping genes are the genes that are absolutely required for the cell to survive. And typically they're expressed at the same level among different species of cells, within different cells in an organism, whatever. You have housekeeping genes in both prokaryotes and eukaryotes. Genes like actin, which make the cytoskeleton, is a good housekeeping gene, or genes involved in glycolysis. All right, so then you have sigma factors for heat shock response. So, only if the cell is exposed to high temperatures would these genes get turned on. Or nitrogen metabolism, right? Only if this bacteria lives in the soil and actually has to fix nitrogen would that species of bacteria have these sigma factors that can recognize those promoters. All right, so up here, this is RNA polymerase in coli. It's got the 2 betas, the 2 alphas, the omega unit, and then here's sigma factor. So sigma factor is what's finding the promoter on the DNA. This is called the holoenzyme collectively, but just RNA polymerase is called the core enzyme. It's made up of all those different subunits. All right, so let's talk about promoters, since obviously the first step in this is going to be the sigma factor recognizing promoters. So promoters that are common to all promoter or specific, so we call specific sequences within promoters, we'll call them elements. Okay? And they come from the term recognition elements. So they are specific nucleotide sequences that proteins might recognize and stick to. 2 recognition elements that are found within bacterial promoters are specifically called the -10, which has this name, the Pribnow box. Or the negative ends the negative 35 region, right? So the more important sequence here that you should be familiar with is the negative 10 sequence, which is TAT, because there's a lot of homology with that sequence in eukaryotic systems. Sigma factor is able to recognize these specific negative 10 and negative 35 sequences. So the reason they're called negative 10 and 35 is when you're looking at the DNA, the transcription is going to start at one specific nucleotide. So that is your first nucleotide. It's usually designated in a figure like this. And going upstream is going in the negative direction. So here's your negative 10 and your negative 35. And then downstream is the positive direction. Okay, so that's why it's negative 10 and negative 35. It's negative 10 and negative 35 nucleotides away from the transcription start site. Okay, so there's +1, negative 10, negative 35. All right, so sigma factor will bind to these negative 35 and negative 10 sequences. So imagine this big protein and it's just sticking over all of this stuff here. It's all down in that area, but specific domains of the protein allow it to recognize these small nucleotide sequences. Now RNA polymerase can come in and associate with the DNA. Okay, so if you look at this as a stepwise process, RNA polymerase will kind of bind the DNA just non-specifically, maybe hopefully close to the promoter, but not at the promoter. Then sigma factor will come in, recognize the promoter specifically, and we call that, it's called the closed promoter complex, because the DNA is still, double-stranded. It's still together by hydrogen bonds in that area, so it's called closed promoter because the promoter is still intact. But in step 3 here, RNA polymerase is now recruited. And it's called an open promoter complex because RNA polymerase is an enzyme that not only can catalyze a piece of RNA, it also has helicase activity. So it can take the DNA apart. So we call that open promoter. That's all it takes to initiate transcription in prokaryotes. Sigma factor binds to the promoter, it brings RNA polymerase over to it, and transcription is initiated. Now, the next step after initiation is elongation, and RNA polymerase will do it as it's by itself. It doesn't need sigma factor anymore. So it will exist as a core enzyme traveling down DNA, down the template strand, and building a piece of RNA. Right? So sigma factor, the subunit, dissociates, and then the core polymerase can continue along to do elongation. Okay, so here we've got, I don't know, I think this figure is actually a little misleading how they have it. So the core enzyme will bind somewhere on DNA, and then sigma factor will find the promoter region, and then it will bring the core enzyme to it. So before the DNA is opened up by the helicase activity of RNA polymerase, it's called the closed promoter complex, but then it becomes a open promoter complex when it's opened up. So it opens up at that +1 site. That's where transcription is going to begin. All right, sigma factor comes off, and then RNA polymerase goes on its own to transcribe the DNA. And that's essentially it for transcription, eukaryotic transcription in terms of initiation and elongation. So the next part is, where does this process end? So how does transcription terminate? And termination of transcription in eukaryotes and prokaryotes is very, very different. In the prokaryotes, there is a intrinsic mechanism. So when you think intrinsic, you think within, right? So this mechanism is just based on the sequence of the DNA itself. And what happens is in the DNA, you have these very GC rich regions. And the mRNA that's produced as a result of those GC rich regions can loop up on itself and form a secondary structure, which is called a hairpin loop. The presence of this hairpin loop, destabilizes the molecule and the whole thing comes apart. The other type of termination mechanism, and I don't know why, but this is not, if I remember correctly, this isn't mentioned in the textbook, is row-dependent termination. Okay, so you do need to know that, it's important. So row-dependent termination requires this row helicase protein, which basically binds to the RNA. It takes apart this, DNA RNA hybrid that's forming as a result of RNA polymerase, and it releases the transcript. All right, so here's row independent. You see is your double-stranded DNA. This is the template strand, that's coding. So RNA polymerase is building as the nascent. That term nascent you'll start seeing a lot. It means the newly synthesized piece of nucleic acid. So that's the nascent strand of RNA. And it's coming up on this rich GC region. You got these GCs on sequence here. The RNA that it produces because of that GC coding allows this thing to pair up and form the secondary structure, which is called a hairpin loop. And think about like the architecture of this whole system here that just throws off the weight of it or whatever, but it makes it very unstable, and the whole transcription machine falls apart. Okay, so that it's intrinsic. It doesn't require any outside proteins to terminate transcription. So we say it's row independent because the other way of terminating transcription requires the row protein, RHO protein, comes in and just disassembles the complex. There's no figure for that one because the author didn't include it. All right, so that is prokaryotic transcription, initiation and termination. Elongation is just the bringing in of new nucleotides, catalyzing that bond. We talked about the bond formation back in chapter 1 or 2. Okay. Uses ribonucleotides with 3 phosphates, which have it at a higher energy state. All right, the next thing is transcription in eukaryotes, and this is a lot more complex. But hopefully I will take it down here to a level that's not too difficult to understand. So it uses RNA polymerases, but unlike prokaryotes, there are a bunch of different RNA polymerases, right? Polymerase 1, polymerase 2, and polymerase 3. The polymerase that we're mostly going to talk about is polymerase 2 because that's the one that transcribes the protein coding genes, right? So that is responsible for making mRNA. But the other polymerases have important roles in generating the ribosomal RNA and also the transfer RNA as well. Specifically, polymerase 1 makes ribosomal RNA, and polymerase 3 makes mostly transfer RNA. All right, remember, those are the 3 main types of RNA: messenger RNA, tRNA, and rRNA. And they all kind of come together at translation, which we're talking about next time. All right, I can mention now, and then we'll talk about it at the end of the chapter. Polymerase 3 also makes a little bit of rRNA, and it makes other non-coding, these POL2 and POL3 makes other non-coding RNAs like microRNAs and siRNAs, things like that. Okay, so eukaryotic RNA polymerase, and we're talking mostly about POL2 here, has a lot of homology to the core enzyme in bacteria. This process is conserved, it evolved, but certainly it's more advanced in eukaryotes. Okay, so the major difference is the requirement that in order to do transcription, you have to have additional factors present, and we call these transcription factors. If you need to do in vitro transcription, it's very easy to do in bacteria. You just need sigma factor, a promoter, and the core enzyme, and you can do it. It'll go. But that's not the same in eukaryotes. You can't do in vitro transcription unless you have additional proteins present in the reaction. So there's about 2,000 different transcription factors in people, and that's a lot. It's a lot of proteins. That's constitutes about 10% of the human genome. So obviously these proteins are very, very important, and they are involved in regulating all the other genes and defining cell types. But of those 2,000 transcription factors, there's only 5 that are required for transcription of any protein. So we call these general transcription factors because all 5 of these are absolutely required to turn on the transcription of any gene. The other 1,995 are used in addition to those 5, to regulate different genes. All right, but you have to have these transcription factors, and they are TF2D, TF2B, TF2F, TF2E, and TF2H. All right, and you have to learn these proteins. Yeah, you just got to practice it. It'll come together. So the first one involved is TF2D. And the ones that follow are B, F, and E, and the last one that associates is H. And they all have specific roles that help bring RNA polymerase, specifically RNA pol2, because we're talking about messenger RNA transcription, together at the promoter. Once all 5 of those transcription factors are with RNA polymerase, this is called a pre-initiation complex. Don't get excited, we're not done yet. It's just a pre-initiation complex. There's an additional important event that occurs. Right? But these general transcription factors position RNA polymerase at the core promoter. All right, so let's look a little bit at the promoter. Remember, the bacterial promoter had just a -10 and a -35. One of those was very similar if you look to this regulatory element called the TATA box. It's the -10. So core promoters will have TATA boxes. They will have initiator elements. They will have BRE elements, which are important in bringing transcription factor 2B to the scene. And additional downstream elements as well. DCE, MTE, and DPE are all important. And so you'll see this on a figure. It'll start to see where these things are positioned. All right, so this is the series of events, though, that involves these general transcription factors in producing the complex that is going to permit any transcription in eukaryotes. All right, TF2D comes in first. It basically has a region that recognizes the TATA box. It has TAF domains on it, right, that bind to these other downstream elements. The second transcription factor to come in is TF2B. It binds to the BRE. TF2F comes in and associates directly with RNA polymerase 2. And then finally, E and H come in and complete the pre-initiation complex. Now, at the absolute basal level of transcription, RNA polymerase and these 5 transcription factors is what you need. But just having these 5 transcription factors and RNA polymerase is not going to give you a very good level of transcription or gene expression. There is an additional big factor, big protein complex that's required in general to get good gene expression. And that protein is called mediator, and we'll get to that in a couple slides, but I just want to mention it. So I've got written down here, CTD is not a TF, but is the C-terminal domain of the largest subunit of RNA pol II. So start getting used to the abbreviations, TF for transcription factor, TXN, which stands for transcription. So CTD is just the C-terminus, right, of RNA polymerase. And it's like this tail that kind of comes off RNA polymerase that is sensitive to kinase activity. So and that's an important step because it's the phosphorylation of the CTT that's going to enable RNA polymerase to take off and undergo elongation. All right, so let's look at this. These regulatory elements aren't that important. INR, DCE, MTE. You don't have to. Some of these specific events are important, especially binding of TF2D. Okay, so here's RNA polymerase. It's the purple thing. This big protein is a transcription factor. It's TF2D, and it contains these different domains, right? Region of the protein that fold up independently, have a defined function. Well, their defined function is to bind regulatory elements on the DNA, okay? So specifically TBP binds to the TATA box. The TAFs recognize the downstream elements because it's a big transcription factor. All right, after TF2D binds, B comes in. And its role is to just kind of enhance TF2D. It stabilizes it. When to remember this, remember the BRE element that's in DNA. So transcription factor B is recognizing that BRE element, but its role is to stabilize TF2D. The last two to join are E and H, right? And think it's the last transcription factors to come in, so it probably provides some signal for RNA polymerase to travel down the DNA and start polymerizing RNA. But when E and H come in, now we have our completed pre-initiation complex. And TF2H is a special transcription factor because it also has enzymatic activity as well. So it's both a helicase and a kinase. TF2H is like the final signal in this process for RNA polymerase to do its job. It unwinds the DNA a little bit at the transcription start site. And because it has kinase activity, it phosphorylates RNA polymerase. And we talked about this before, phosphorylation, good post-translational modification that activates proteins. So where it phosphorylates is this, like the tail of RNA polymerase called the C-terminal domain. Once you get all these phosphates on there, now RNA polymerase take off and move down the DNA. Okay, a little more about TF2H. It's a serine kinase, okay, which means specifically it phosphorylates the serine residues on that CTD. It's looking for this sequence. You don't have to memorize this. No, it's a serine kinase. All right, this is like the molecular switch, and phosphorylation, glycosylation, a lot of those as we move forward in this class, you're going to see is like the on switch for a process. And it's very true in eukaryotic RNA polymerization. All right, so that pre-initiation complex, you can get transcription of genes, but it's not at a very high level. There is a large protein that can come in, though. It can associate with these transcription factors we've been talking about. It can associate with RNA polymerase, and it can associate with other transcription factors to really increase expression levels, right? You think about what a mediator is, right? In a divorce, right? It's trying to work both sides. And that's what this protein is doing, essentially. It's balancing out other transcription factors that can come in and allow hyperregulation of this gene. Okay? So it works with activators, it works with suppressors. And all in all, it can increase transcription by stimulating the kinase activity of that final general transcription factor that comes into the complex, which is TF2H.
Is this the polymerase too?
It's polymerase 2. That's right.
That's all this.
Yeah, and so it's a really good question because pole 1, as we're going to see, but I'm not really requiring that you learn. Pole 1 and pole 2 have very specific different transcription factors that are involved in their regulation. So yeah, this is all pole 2. It's a good question. Okay, so there's mediator. It's like a big cap that sits on TF2D and wraps around so that holding on the general transcription factors. But there's active regions of the mediator out here. Something we'll learn about a little later on. DNA can have enhancer regions. Enhancer regions are regions way down far away from the promoter site that can actually cause the DNA to bend and have transcription factors on it, and those interact with mediator protein. When you look at like how the transcriptional machinery is arranged in the nucleus, these mediator proteins are really large, and they make up what are called transcriptional hubs that are regions of high transcription activity in the nucleus. And we'll get to that in the nucleus chapter. It's kind of interesting to see that. All right, so after TF2H phosphorylates the CTD domain of polymerase 2, it can take off. It brings with it TF2F. You don't need to, we're not going to get into that though. Okay. So now it's doing elongation. We have initiated transcription in eukaryotes, and as you can see, it's a lot more complex than just sigma factor finding the -10 and -35 in prokaryotes. Okay, so how does termination occur in eukaryotes? Well, the good news for you is we really don't know very much, so not to learn it, but we have some ideas. Okay, so one idea is that CTD phosphorylation, remember the tail of that RNA polymerase, will actually recruit enzymes to the nascent RNA that start breaking it up, right? And it breaks it up at a specific region. So right now we're talking about the reduction of a pre-mRNA, which is the nascent messenger RNA. But something that's going to happen after it gets produced is it's going to get modified, and it's going to have a 5' cap, which we'll talk about, which increases stability and helps direct it to the ribosome. And it also has a polyadenylation sequence, which is the string of A's that exist at the end of it. So that string of A's is a signal for transcription to definitely stop. Right? It starts destabilizing the mRNA that's getting produced. Right. So, that in addition to CTD phosphorylation is a good indicator that poly-A signal, or good indicators that transcription is stopping. All right. So there you can see polymerase 2 is starting to approach the polyadenylation signal. And that CTD by doing what are called pull down assays, right? So you have purified proteins out of the cells, and you have an antibody against certain proteins, and then you can pull that in a tube, and any proteins that are stuck to it, you can actually find out what those are by running them out on a gel and sequencing it. And pull down assays have found that these endonucleases, specifically RNA endonucleases, tend to stick to the phosphorylated CTD domain of RNA polymerase. So these endonucleases, the red thing, will come in now and just cut off the nascent growing strand of RNA where that polyadenylation signal is. Okay, so here's where this starts to get annoying. If you haven't been annoyed already. There's not just RNA pol 2. There's more to the story. For example, RNA polymerase 1 is what's responsible for making most of the subunits of or the parts of the subunits of the ribosome. So just to refresh your memory, because this, looking at this, which is what RNA polymerase makes, most of these, won't make much sense. So where are these fragments of RNA going to? And that's in the individual subunits that are found in eukaryotic ribosomal subunits. So the mammalian ribosome, which is termed ADS, the S stands for Svedberg units, which is like the distance it'll migrate in a certain type of centrifugal gradient. The ADS is made up of a combination of a large and a small subunit in mammals. The 60S and the 40. What's 60 plus 40? 100, right? So that doesn't add up. And that's because it's not like nucleotides. It's a migratory distance in the tube. So it doesn't have to add up. It has to do with other properties of the molecule than just the size. All right, but the whole thing is 80. It's made up of a 60 and a 40. And specifically, the 60 is made up of 3 fragments, right? You should start to become a little generally familiar with this because it's not the last time we're going to see this stuff. When we get into translation, you're going to see these units come up again. And the 40S is made up of primarily that 18S transcript, right? Remember, the ribosome is made up of protein and RNA. So here we are explaining where does the RNA come from? The RNA comes from RNA polymerase 1 generating these transcripts. And the only one that's kind of an exception, super nerdy, you'll remember the 5S is kind of an exception. It's actually generated by RNA pol 2. Right, but the 28S, 5.8, the 18S are all generated by RNA pol 1. Okay, these are the components of the ribosome. Oh, did I mix that? I might have said that wrong. Yep, so RNA pol 1 produces the 28, the 18, and the 5.85S. Okay. Now, how do you get the 28, the 18, and the 5.85S? They actually come from a larger transcript, a continuous transcript that is size 45S. And why this is important is because we're going to start now getting into the processing of RNA. Just because you've produced RNA as a result of transcription, you're not done. The RNA needs to be processed. The mRNA is going to get processed, cap and tail, but rRNA and tRNA are also processed, right? The processing of rRNA is the development of going from a 45S into these smaller pieces. There's a figure that kind of shows you this. You don't need to know any of these regulatory elements or these transcription factors with RNA pol. Okay? Just know the elements and the factors for RNA pol 2. The purpose of this figure is just to show you how one continuous pre-transcript is made and that there is processing events that make the smaller 18, 5.8, and 28 coming together to make the large ribosomal subunit. Okay, so RNA pol 3. So pol 1 makes most of the rRNA. Pol 2 makes the mRNA. So what makes the tRNA? RNA pol 3 makes the tRNA. Okay, so not much I'm really requiring that you know here. Just know that the process of RNA pol 3 making the tRNA has its own promoters. It has its own transcription factors, like you can see SNAP and TF2C. Those are pretty new. We haven't talked about those yet. It shares TF2B with, RNA pol 2, but this RNA pol 3 has its just own processes that are used to generate the tRNAs. What we'll talk about in the next section is the processing of tRNAs, and that is a little more significant, I think, than just the transcription of it. So that's as far as you need to take that. Okay. All right, you guys take 2. What time is the class over? 12:15 or something? 12:30? Hopefully we'll get out. Maybe we'll get out a little early. Take 2 minutes and we'll do this last section and then be out of here. You got to see me for a week, right? Monday. Monday you do. Well, not speaking about this stuff, right? Oh, get a week break. Good chance we might have a quiz on this though on Wednesday. So please keep that in mind. Yes, ma'am.
If there is a quiz or
What do you mean you're missing? Pam?
the email you sent my interview.
Oh,
On
yeah,
Wednesday.
that's the excuse. So just get with me when you come back. Yeah.
Okay. So take it Monday or
Yeah, that's fine.
if there.
Yeah, you can do that. No, I can't do that. Just do it when you get back. All right, guys, you ready? Let's get this done. RNA processing and turnover. Okay, so I've kind of introduced you to what RNA processing means. After you have the pre-mRNA or the pre-rRNA or the pre-tRNA, that's got to get processed, right? mRNA is going to get cap and tailed. We know the rRNA, that pre-transcript, gets cut. And then turnover as well. So what does that mean by turnover? Well, how, does the RNA linger in the cell, right? Because, if you have an mRNA present, then it can go to the ribosome and a protein can get produced. And the mRNA comes off the ribosome and then it can be translated again. So the more mRNA copies you have in a cell will dictate a higher level of gene expression, but also how well those RNAs stick around the cytoplasm will also, will also affect your level of gene expression. So when we talk about RNA turnover, we talk about, the RNA getting degraded, being regenerated, things like that. Okay, so in general, processing of these things, tRNA and rRNA, it's the cleavage of larger precursors, like we saw with the rRNA. But there are also chemical modifications that can occur, specifically at the ribose. So two of them, methylation and the production of these like pseudo-uridine molecules. We'll see that in a little bit. And then the mRNA, we know it gets a cap, it gets a tail, and it gets spliced, right? The introns are removed and the exons are annealed together. All in all, this is ensuring that the RNA molecules are stable, that they're effective, that they function correctly, they're ready for the next step, translation or assembly or whatever. All right, we already talked about this. The 45S precursor is cut into 3 smaller pieces that are come together to make up, the large subunit of the ribosome. But the rRNA can also be chemically modified. Right? The ribose can have a methyl group onto it, or you can get its modified form of uridine. I thought there was a figure. Yeah. So here's uridine isomerization here, ribose methylation there. All right. So, the full function of those modifications we're not going to get into, but they are important in the maturing of the rRNA. Now, the molecules that do this, though, are important. And so, you think, where is this occurring in the cell? Right. So it's mostly occurring so this has to do with rRNA, right? So where do ribosomes get assembled? Where do ribosomes get assembled? It's in a little central area of the nucleus called the nucleolus. So nucleolus is responsible for ribosome assembly. And so the proteins that are found in the nucleolus are abbreviated small nucleolar, stands for NO, ribonucleoproteins, or SNO RNPs. All right, these are the SNO proteins. And I'm not requiring that you know all the snow proteins. I'm not, we had a snow lab down the hall from us, and I don't know anything about these snow proteins, and I really don't care. But they do some important stuff. They help process the our RNA. So you basically have ones that can do the methyl, that act as methyl transferases. They add methyl groups to the ribose, or you have ones that are capable of doing this uridine isomerization. All right, so here you've got a collection of these snow proteins that are methylating ribose on our RNA that it's maturing, collection of proteins that are making this uridine isomer. Okay, just further processing. All right, processing of tRNAs, I would like you to look at though a little more closely. So, and just because it has these very unique enzymes that are involved in its processing that are really commonly used in molecular biology, that include RNase P and RNase H. The tRNAs, when they get transcribed, they exist as a pre-tRNA. And then after that, eventually it has to fold up into that clover leaf structure, right? So it'll look something like this. Right? The tRNA, when it's working in the ribosome, looks something like this. But if you denature it completely, it should look like this. So remember, this is the anticodon region. This is the arm, and then this is the amino acid that's tagged onto it. Okay. So there's two cleavage events that occur to the tRNA. You get a 5' cleavage and you get a 3' cleavage. And one important enzyme here is this RNase P, which is a ribozyme. Ribozymes, remember, are pieces of RNA that have catalytic activity. And RNase P cuts the 5', and then just like the typical old RNase, which is called RNase H, cuts the 3'. Those are two steps. And then the third step is you get the CCA that's added to the 3' end. And the reason that's important is because there are enzymes now that will charge that tRNA by adding an amino acid to it. And it can only do that because that CCA sequence is there. So this helps create a very stable tRNA structure that can be used in translation. All right, so first step up there is 3' cleavage, sorry, 5' cleavage by RNase P. Then you get cleavage of the other end. And then you get the CCA added on here, right? It's the 3' end that's going to be that the amino acid comes onto. Memorize that. And then there's a whole bunch of base modification events that can occur. The uridine isomerization, methylation, that's what these are indicating here. And that's a real intricate part of tRNA synthesis and maturation that's still being completely understood. Okay, so that's rRNA and tRNA. We're done with that. And let's move on to mRNA, which are the actual transcripts that code for genes. So in order to go from a pre-RNA to a mature RNA, you have to give it a cap, you have to give it a tail, and you have to splice it. Right? And splicing definitely is the most important, but the most implicating of those three events, because it's the splicing that allows the protein diversity, because you can have alternative splicing that occurs. Right, but the cap and the tail are primarily there for stability reasons, and it also helps the next process, which is translation. Okay, so we've got our DNA, which is transcribed by RNA pol 2. We get a pre-mRNA that's made. It includes both the exons and the introns. All right. First thing that occurs, it gets a cap. Right, we'll look at the cap more closely. It's like a modified guanine that's methylated. It gets a tail. which is a series of A's that come after the pre-mRNA. At the 5' end, this is the 3' end, and it gets spliced. The splicing is the removal of the introns. All right, let's look at the cap. So the cap is a unique, situated, modified guanosine that sits at the 5' end. The reason it's uniquely situated is the type of linkage that occurs there. So all the linkages we've been talking about are always 5 to 3, right? Phosphodiester bonds are 5 to 3. But the G, because it's methylated, it produces this 5' to 5' linkage. So here is your mRNA, the 5' end of it. And the cap is a guanine that is methylated. And it's linked to the 5' end of the mRNA at its 5' end. And that doesn't make sense, shouldn't make sense to you, because it should be the 3' carbon that is linking to it. But that's what's unique about this 5' cap. And it actually, it changes the architecture of it so that that cap like sits off of it. It is a little separated, a little further away from the mRNA. And it's the cap that's going to allow it to get translocated to the ribosome so that it gets translated. Okay, so the cap has a methylated guanine attached through this unique linkage, 5' to 5' linkage at the 5' end of the mRNA. All right, poly-A tail. We're almost done, guys. Poly-A tail. The poly-A tail is added through the enzyme poly-A polymerase. Poly-A polymerase will add 200 A's at the 3' end. But how does it know where to do that? Well, first, something needs to come in and cut the RNA at the correct location. So there's endonuclease that is able to cut the mRNA. But the endonuclease has to know where to cut the mRNA. And the way it knows that is through a signal sequence, all right? And the signal sequence is its unique AU sequence that is found in these very GU-rich regions. So the endonuclease comes in, it finds the signal sequence, right? Cuts a little down of it. Now poly-A polymerase can come in and add a string of 200 A's to it. Okay. That GU-rich element is basically also the signal sequence for the termination of transcription. The functions of the poly-A tail, the biggest one is to protect it from degradation. So if you ever work with RNA in the lab, it's a real pain to work with. RNA is easily degraded by these well-conserved, in terms of evolution, enzymes called RNases. Right? RNases are enzymes that degrade RNA. And they're very, very abundant in the cell. They survive on surfaces. When you're talking, you're like letting them out. I mean, it's a very easy contaminant in the lab. And they're present in cells as well, and they will break down RNA. The presence of that poly-A tail will prevent that. So it stabilizes the RNA and prevents degradation through RNase. It's also important in getting the mRNA out of the nucleus to the cytoplasm. And it has some roles in kind of stabilizing the whole translational complex. All right, so how is the poly-A tail added? So you can see the recognition element there, the AAT, AAA. You can see the downstream, that rich GU thing. First thing that has to happen is the endonuclease comes in and it cuts it at this specific CA residue, CA sequence here. And then after the endonuclease cuts, poly-A polymerase can add the string of A's after that. Okay, and now you have a stable and finally almost mature messenger RNA. There's still one more thing we have to do. What is that? We've capped it, we've tailed it, and now we got to splice it. Okay. Splicing is the most intricate of all of these three, and it takes up about probably 6 pages or so in your chapter, and author actually does a pretty good job of explaining it all. We're not going to get into it too deep, all right? In fact, this is about as bad as it's going to get, but there's a lot to it that we're not going to cover, and that's okay. If you take molecular biology, you'll get into more of the details. All right, so what is splicing? All in all, it's removing the introns and then sticking together the exons. Now you finally have a mature mRNA. So this is a type of post-transcriptional modification, right? We already have our pre-transcript, how we're modifying it, cap tail splicing. The protein complex that is doing the splicing is called the spliceosome. It's a combination of both protein and not small nucleolar, those were the SNO, SNO proteins, but small nuclear ribonucleoproteins. Okay, SNRNPs and proteins make up the spliceosome. It's a big complex. And you can think of these like SNRNPs and proteins all like little robots coming on to, work on this mRNA and cut out the introns. What this enables, though, ultimately is alternative splicing, and now you can get a real good protein diversity. You get a bunch of splice variants just from one gene. And, maybe all those splice variants, they probably have a very similar job, but they can have subtle differences that enable that complexity in the cell. All right, so this is just kind of showing how this process was discovered. Basically, here you've taken like from just a plasmid and you've cut out, a segment of DNA that has both exons and introns, and you mix it with a nuclear extract, and all of a sudden now you have a shorter piece of DNA. So there must be something in that nuclei that is cutting the DNA in a very orderly fashion and then ligating it back together. And that's how the SNRNPs, the spliceosome, was discovered, by doing that. All right, so what is the mechanism? Well, you have 2 splice sites, right? The splice sites are where basically exon meets intron. You have a 5' site, you have a 3' site, 3' splice site, and you have this other component that actually enables the mRNA to loop up together, and it's called a branch point, and it's a specific adenine nucleotide that allows this loop to form, and it's called a lariat loop or lariat structure. All right. All this is coming together in sequence. This process is very well understood, and all the proteins have been recognized and crystallized. So we got 2 figures on this. This first figure is just going to show the generic sequence, like how the lariat loop forms. The second one kind of shows the important proteins that come together to form the spliceosome. And then that's it, right? The rest of the chapter or the rest of the section you can ignore on this. It's just getting a little too deep, I think. Okay, so here we've got 2 exons and an intron in between it. And well, obviously you're going to have a splice site on both sides of the intron. So you got your 5'1" and your 3'1, and that branch point is some A that's found in, semi-middle region of the intron. All right, you get proteins that cleave the 5' first, right? Then you get proteins that bring the loop around at that branch point. You can see the A branches with the G. It's kind of a unique type of nucleotide interaction. And then you get cleavage at the 3' site. Okay. And now you have your intron, which is looped up, taken out. Actually, that looping is important for degradation of it. And you have your exons that have been brought together. And that religating essentially is the last step. Process the mechanism of splicing. Okay, so the splicing proteins are these U proteins. And, I think when they started devising this mechanism, when they were labeling the proteins, they were very nice and just gave them simple names. So they're all U proteins, and they go from 1 to 6, and that is the order that they operate in. So U1 does the 5' slice, does the 5' cleavage. U2 sticks to the branch point, the adenine. And then U4, 5, and 6 come together, and now you have the spliceosome formed. Okay. And that's the assembly, right? So in order to do the rearrangements, like in order to do the 5' cutting and to form the lariat, those proteins are involved in it, right? So, you know, like U1 and U4 have to leave. There's a step where they come off and the lariat forms. And that's about as complex as we're going to take this, okay? But this is a really big protein complex, you know, and they're operating in the nucleus, right? All the maturation that occurs for the RNA, the tail, the cap, and the splicing is all occurring in the nucleus. All right, 2 more slides here. So that's RNA processing in terms of like the modification of RNA. But then you also have to think about RNA turnover, right? So RNA turnover is related to the levels of RNA that you have in the nucleus, have in the cell at the time. There's also processes that can occur that can actually edit the RNA. So a very common form of editing involves transitioning from an A to an inosine. And you know, it's similar to an A, but it's not going to get translated the same. It's another form of mutation. And then degradation of the mRNA through enzymes like RNases, like I talked about before. So there's 2 ways to break up the RNA. You can either take the cap off, or you can start breaking down the tail. It's basically what they call the poly A tail shortening, deadenylation. And here you've got deadenylation occurring, decapping occurring. Both of them, removing the cap or the tail, makes the RNA molecule sensitive to nucleases, exonucleases. This is an important step and process related to gene regulation. Okay. All right, guys, a lot today. Again, it's all new material. Could be most of it. Please start reviewing it. You know, might have a quiz on Wednesday. See you guys on Monday for the Rubisco lab.
Trying to memorize. Okay, everybody,
Practice makes.
Outside, there's food, either
Ooh.
a hot dog or a veggie dog with chips and a drink, so just grab a ticket. Oh, go get free lunch.
There you go.
It's Constitution Day. How do I find? You're welcome.
like team